The classical pathway is one of the three primary activation pathways of the complement system. The classical pathway is initiated when antibodies, usually immunoglobulin G, IgG, or immunoglobulin M, IgM, bind to specific antigens on the surface of pathogens. This binding event creates an antigen-antibody complex. The C1 complex is a large protein complex composed of three subunits, C1q, C1r, and C1s. When the C1q component of C1 binds to the Fc region of IgG or IgM antibodies in the antigen-antibody complex, it undergoes a conformational change. The active C1r fragment then goes on to activate C1s, which is also a protease. Activated C1s cleaves the next component in the cascade, C4, into C4a and C4b. C4b, in combination with another complement protein called C2, forms a complex known as C3 convertase. This complex has enzymatic activity and can cleave the central complement protein, C3, into C3a and C3b. C3b can go on to bind to the surface of pathogens, marking them for phagocytosis, engulfing and destruction by immune cells, through a process called opsonization. Moreover, the classical pathway can continue to amplify the cascade by forming additional C3 convertases, resulting in more C3b molecules and enhancing opsonization. Further cascade activation, the cleavage of C3 into C3a and C3b leads to downstream activation of other components of the complement system, ultimately leading to the formation of the membrane attack complex, MAC, and the release of anaphylatoxins like C5a. The MAC attack is initiated when the complement protein C5 is cleaved into C5a and C5b. C5b is the key component that starts the formation of the MAC. C5b quickly associates with C6 and C7, forming a complex known as C5b67. This molecule attaches to the lipid bilayer membrane of the target cell, which is often a bacterial cell. This binding promotes the recruitment of additional complement proteins, specifically C8 and multiple units of C9. This molecule form a tubular structure that spans the lipid bilayer, causing the bacterial cell to lyse.